Hello friends, welcome to BMH Learning. This video is regarding Spindle Assembly Checkpoint. The Spindle Assembly Checkpoint, also known as the Metaphase to Anaphase transition, is a cell cycle checkpoint during mitosis or meiosis that prevents the separation of the duplicated chromosomes until each chromosome is properly attached to the spindle. To achieve proper segregation, the two kinetochores on the sister chromatids must be attached to opposite spindle poles. Only this pattern of attachment will ensure that each daughter cell receives one copy of the chromosome. The defining biochemical feature of this checkpoint is the stimulation of the anaphase promoting complex by M-phase cyclin CDK complexes, which in turn causes the proteolytic destruction of cyclins and proteins that hold the sister chromatids together. The beginning of metaphase is characterized by the connection of the microtubules to the kinetochores of the chromosomes, as well as the alignment of the chromosomes in the middle of the cell. Each chromatid has its own kinetochore, and all of the microtubules that are bound to kinetochores of sister chromatids radiate from opposite poles of the cell. These microtubules exert a pulling force on the chromosomes towards the opposite ends of the cells, while the cohesion between the sister chromatids opposes this force. At the metaphase to anaphase transition, this cohesion between sister chromatids is dissolved, and the separated chromatids are pulled to opposite sides of the cell by the spindle microtubules. This was all. Thanks for watching.